As Randy mentioned, the topic of my talk today, as well as my thesis, has been these two G-protein coupled receptors. So if it's been a little while since you thought about a G-protein coupled receptor, or if you've never actually considered one, that's all right. I do want to tell you, though, that G-protein coupled receptors, which are abbreviated GPCRs, are the largest class of cell surface receptors in the human body. Being a cell surface receptor means that they bind a ligand, such as a neurotransmitter, on their extracellular side. That binding of the ligand induces a conformational change within the receptor. And when the receptor changes its conformation, it then induces intracellular signaling within the cell. And because these are G-protein coupled receptors, that intracellular signaling is mediated through a G-protein. So G-protein coupled receptors, because of their wonderfully widespread expression, as well as their modulatory ability for signaling, are excellent therapeutic targets. In fact, anywhere from 30 to 50% of the drugs on the market today target a GPCR. And GPCRs are also responsible for the signaling for a number of neurotransmitters that we know and love, like dopamine or norepinephrine. So I guess the way that I've presented this sounds good, right? Everything's wrapped up in a neat little bow. There's a very clear pathway, ligand binds, receptor signals, everybody's happy. But the problem is that there's a huge gap in the field. And that gap is the fact that there are still over 100 so-called receptors that do not have a ligand. So there's nothing binding to this extracellular space that we know of, which means that we don't know how the receptors signal and we don't know what their function is. And these ligandless receptors are referred to as orphan GPCRs. And orphan GPCRs are just a really hot topic in neuropharmacology. One, because it's thought that they might contribute to novel therapeutics for neurological disorders, and two, because it's thought that they may actually provide one of the mechanisms for some sort of neurological diseases. So, as you might have guessed, two receptors that have attracted particular interest are the subject of today's talk, and that is GPR37 and GPR37L1. Now, given the similarity of the receptor names, I'll probably just start calling them 37 and L1 to help keep everything the same, or kind of clear. So if we don't know what type of ligands are activating 37 and L1, and we don't know how they signal, what do we know about them actually? Well, what we know about the receptors, we mainly know about GPR37. And what we know about 37 comes to us from the context of Parkinson's disease. So as everyone in the room knows, I'm sure, Parkinson's disease is a neurodegenerative disorder. It's marked by a progressive loss of dopaminergic neurons. Now, there are a number of potential contributors to the disease, a number of potential causes, but it's believed that one of the kind of major contributors is the dysfunction of this ubiquitin proteasome pathway. And this is actually how GPR37 was first discovered. So GPR37 was discovered as a substrate for an E3 ubiquitin ligase called Parkin. And if Parkin is dysfunctional, or actually I should tell you also that because 37 was discovered as a substrate for Parkin, the alternative name for 37 is the Parkin-associated endothelin-like receptor. And I also want to point out that you should be very grateful that I'm calling it 37 because that shaved a solid 5 minutes off this talk. GPR37 as a substrate for Parkin actually means that if Parkin is dysfunctional or mutated, as it happens in a type of juvenile Parkinson's disease, that means that GPR37 can now misfold and accumulate in the cell. And if GPR37 accumulates in the cell, that leads to cell stress and cell death. So that little nugget of information might actually make you say, well, clearly this is a detrimental protein, let's just inhibit it. Don't even worry about its signaling, just get rid of it. This is actually not the case. So if we were to get rid of GPR37, what do we do with GPR37 knockout mice? If you take GPR37 out of the normal healthy mouse, you actually see some severe perturbations in the dopaminergic system. 37 knockout mice have a lower dopaminergic tone, and they also have alterations to their DAT, their dopamine transporter, and also dopamine receptor functions. So this indicates to us that GPR37 actually does something actually incredibly important in the brain, and it gives all the more reason for us to try and find a ligand for that ligandless receptor. So when I joined the project, that was our goal. And we set out to do this by first establishing some criteria that we thought a ligand for 37 or L1 should have. So the first criteria being that since 37 and L1 are so closely related, we knew that they were probably going to bind the same ligand. We also knew, based on the structure of the receptor, that these two receptors should bind a peptide ligand, so that narrows the search even more. we knew that since they are GPCRs, we were going to have angiogenic signaling effects mediated through G proteins. Additionally, I've told you about the dopaminergic component that we looked at. And finally, we thought maybe, just maybe, it would be important if the ligands were actually expressed in the same brain regions as the receptors. That seemed like a good criteria. After establishing our criteria, we went through and thought about what we know about GPCRs to try and establish what sort of output we were going to look at for receptor activation. And we finally settled on an output that is known as internalization. So internalization is a very common property of GPCRs. When a ligand binds to a receptor here, the receptor undergoes desensitization and then it rapidly internalizes from the cell surface. And that's something that's actually very easy to measure. So we've got our criteria, we've got our output, and we actually started screening our peptides to see if any of them were able to induce the internalization of 37 or L1. You can see here that 4 out of the 5 peptides that we have screened induce absolutely no specific internalization of the receptors. But one peptide was able to induce the internalization of both 37 and L1. And that peptide is called prosaptide. So I was thrilled, so excited. This was such a cool finding. But of course, my next question was, what's prosaptide? What is it? So it turns out that prosaptide, being a peptide, like I told you, is actually this 14 amino acid peptide that is derived from the neuroprotective factor prosaposin. Now, prosaposin and prosaptide have their own literature that dates back about 20 years from when prosaposin was first discovered. And all the literature on prosaposin and prosaptide identify them as neuroprotective and glioprotective proteins. And they also say that prosaposin and prosaptide actually exert their protective effects through an unknown GPCR. So, real exciting. 2 protective effects that I want to point out, because they'll come up later, are the ability of prosaposin to enhance peripheral nerve repair, and also the ability of prosaptide to rescue dopaminergic neurons from an MPTP insult. So we showed that the ligands, yeah?
What's an MPTP?
Oh, excellent question. I should have explained that. So MPTP is actually a toxin that's used to mimic a Parkinson's disease state. So it's kind of environmental toxin, gets in, messes up dopaminergic neurons. So that's an excellent question. Thank you. And anybody else, if you have questions, please interrupt. So after establishing that prosaptide and prosaposin were able to induce the internalization of receptors, which is a good start towards looking at their activation, we decided to look and see if we could identify other characteristics and hallmarks of a GPCR ligand binding to, or a GPCR ligand and its receptor. So the first one we looked at was binding. Here you're able to see that we added prosaptide to beads, and then we used those prosaptide on those beads to actually pull down GPR37 and GPR37L1. But those prosaptide beads were not able to pull down other GPCRs, such as the alpha-1A or beta-1 adrenergic receptor, the dopamine D1 receptor, or even the endothelin B receptor. And this is exceptionally important because the endothelin B receptor is the most closely related receptor to 37 and L1. So this indicates to us that we have a very specific interaction between the prosaptide ligand and 37 and L1. Additionally, we knew from the prosaptide literature that prosaptide was able to be pertussis toxin sensitive. And so pertussis toxin, in addition to being a bacteria that causes whooping cough, actually is able to inhibit GI proteins. So we knew that prosaptide was likely acting through a GI protein coupled receptor. And the hallmark of the GI protein coupled receptor is its ability to inhibit cyclic AMP. So here you can see that we did that. We showed that GPR37 and GPR37L1 are actually able to mediate the ability of prosaptide to inhibit cyclic AMP, suggesting that they are GI coupled receptors. Also, our experiments have all been pertussis toxin sensitive, and we were even able to use prosaptide to stimulate the ability of the G-alpha-I protein to bind to our 2 receptors. So everything so far points to prosaptide and prosaposin binding to 37 and L1, stimulating the receptor signaling through GI proteins. But what we were truly interested in was the ability of the receptors to mediate a stimulation of phospho-ERK. So ERK phosphorylation is one of these potential downstream pathways of GPCR. And when ERK gets phosphorylated, it becomes activated, and activated ERK is able to mediate a cell survival pathway. And more importantly for us, prosaposin and prosaptide have both been shown to actually activate phospho-ERK and do so in a pertussis toxin sensitive manner, And it's also been further shown that the activation of phospho-ERK by prosaptide or prosaposin actually results in cell protective effects. So we wanted to see if our receptors were actually mediating that ability of prosaptide or prosaposin to induce phospho-ERK signaling. To do this, we started with HEC293 cells. So HEC293 cells are an immortalized cell line. They normally, as you can see, are not at all responsive to prosaptide. If you add prosaptide, you don't see any change in ERK phosphorylation. We then added in the receptors through transfection, and you can see that if we add in GPR37, you suddenly now get a robust increase in ERK phosphorylation if prosaptide is added to the cells. The same thing is true for GPR37L1. So both receptors are able to mediate the ability of prosaptide to stimulate ERK phosphorylation in transfected cells. However, I do want to point out that so far everything I've showed you, we've taken cells that normally don't respond to prosaptide, we add in the receptors, and we get a prosaptide response, whether it's signaling or binding or things like that. However, we also wanted to see if the reverse was true. We wanted to see if we could take cells that normally are very responsive to prosaptide and express our receptors and try to remove our receptors to see if we could subsequently remove the cell's responsiveness to prosaptide or prosaposin. And for our model system, we chose cortical astrocytes. Cortical astrocytes express both receptors, they respond to prosaptide, and most importantly, they are very amenable to siRNA knockdown, which allows us to remove each receptor individually. So using our cortical astrocytes, we repeated our phospho-ERK experiments, and you can see here that normally prosaptide induces this nice robust increase in ERK phosphorylation. However, if you were to remove 37, or both receptors together, you completely remove the ability of prosaptide to signal to phospho-ERK in cortical astrocytes. And the same is true of prosaposin, that full-length protein. It's interesting to see that if you remove L1, you don't get any change in ERK phosphorylation in the astrocytes, even though our transfected cells showed us that you really could actually induce ERK phosphorylation with L1. There are a couple of different reasons for this that we're working on in the lab. One is that even though both receptors are expressed in cortical astrocytes, GPR37 is far more highly expressed, and therefore may be mediating much of the phospho-ERK signaling. Another possibility is that even though L1 is capable of mediating phospho-ERK signaling with prosaptide, it may actually have an alternative pathway that it prefers in the cortical astrocytes. So this shows kind of that we can get signaling. We know that if you add in the receptors, you get signaling where you didn't before, and we know that if you take away the receptors, you lose this ability for phospho-ERK signaling. But really kind of the true test of trying to identify a ligand is to see if you can actually change a functional response. And what better functional response to look at for a neuroprotective protein or a glioprotective protein than the ability to protect glia. So we used our same cortical astrocyte setup with siRNA knockdown, and we stimulated those cortical astrocytes with hydrogen peroxide and measured the ability of prosaptide to protect the astrocytes against the oxidative cell stress and cell death that hydrogen peroxide induces. And you can see here that for both prosaptide, this short amino acid protein or peptide, as well as the full-length neurotropic factor prosaposin, you normally get a nice protection effect of the two, of prosaptide and prosaposin. However, if you were to remove 37 or L1, or both receptors simultaneously, you obliterate that protective ability of prosaptide and prosaposin. It's gone. We do think that the phospho-ERK signaling is mediating the protection through GPR37. However, it's incredibly likely that L1, which you'll notice if you remove it, it actually removes the protection, is actually an alternative signaling pathway that I proposed with the cortical astrocytes. Yes?
How do you measure protection?
Measure protection? Excellent question as well. So we have we have a vehicle-treated plate, so we can see kind of the basal. We're actually looking at LDH release, just as a further information. So we look at this basal LDH release. We look at the LDH release that is caused by the stimulation of the hydrogen peroxide in the separate well, and then in other wells we have hydrogen peroxide plus prosaptide and all very different conditions. And then we take the percent cell death that we can calculate with the LDH release, and then we compare that for the group that was treated with prosaptide versus the group that was not treated with prosaptide. And using that, we calculate the amount of protection that prosaptide is able to give. Yeah?
Is your phospho-ERK mediated by G protein dependent beta gamma signaling, or do you think it's G protein independent, like through arrestin mediated signaling?
So, an excellent question. We actually haven't looked as much at arrestins as we should, I'm sorry to say. I do think it is mediated by kind of the G protein beta gamma because our phospho-ERK is pertussis toxin sensitive, which suggests the involvement of the G alpha I protein and beta gamma as well. Really good. Are there any questions? Okay, excellent. So, what I've shown you today is that we have identified that prosopocin, that endogenous neurotrophic factor, as well as the neuroprotective peptide prosaptide as ligands for these orphan receptors GPR37 and GPR37L1. We further identified that 37 and L1 are able to mediate the protective actions of prosaptide and prosopocin in astroglia. So for future directions for this project, we actually want to take advantage of the incredibly collaborative environment here at Emory. And we're working with Mike Cottle's lab to assess whether or not our receptors are mediating the protective ability of prosaptide, or the ability of prosaptide to protect against those MPTP insults, so protecting dopaminergic neurons against Parkinson's-like insults. And we'll be doing that both in cell cultures, which I'm doing now, and also in vivo studies. And we're taking advantage of our newly acquired receptor knockout mice to see if we can actually identify the receptors as that link for prosaptide protection. Additionally, working with our English's lab, we are going to assess whether or not our two receptors are able to mediate the other well-known ability of prosopocin to actually facilitate peripheral nerve repair following injury. So with that, I want to thank the people who've really driven this work forward. First of all, my lab, who on occasion looks normal, and we're sure to take a picture when it happens. I specifically want to thank my outstanding mentor, Randy Hall, whose creativity and mad science skills never fail to impress me. I want to also thank Steph and Michelle, who have been an everlasting source of guidance and support with the project. I, of course, want to thank my fabulous committee, who have provided a lot of advice and insight over the years, and especially my really fantastic collaborators, Art and Mike, as well as Josh working in his lab, for just being a great source of information or help or anything that I needed. I also want to point out that although we've had fantastic rotation students who have come through the lab, two in particular have really contributed to this project. So for future graduate students, your rotations are not wasted, they're greatly appreciated. And then of course I want to thank the Neuroscience program. Like, this is such a creative and collaborative environment. I really couldn't imagine doing a project like this anywhere else. And I've noticed over the years, all Frontier speakers tend to have like a secret hidden platform that they want to talk about where they have your undivided attention. And as Randy alluded to, my platform is the availability of teaching fellowships at Emory. And these fellowships are a great opportunity, but I specifically want to point out to all the new recruits that the Neuroscience program is always strongly represented in these fellowships. And I do believe that's because the program encourages mentors like Randy, who embrace this idea that science doesn't just belong in a lab or in a conference, but also in a classroom and in a community. So with that, I'm going to step down off my soapbox, and I will take any further questions you have.